last of all. So thank you very much for the introduction. As was said, my name is Ashleigh Geiger. I'm a PhD student at SAHMRI. From SAHMRI, the cheese grater. Like Natalie, you have a SAHMRI sandwich here tonight. Very fortunate. And I'm here to tell you how science could change your fate. That's right. Destiny-altering science, right here, right now, all for the low entry price of $25 a head. Listening to my talk tonight is a little bit like going and reading your horoscope, except this is real. I'm not going to be up here like, beware unexpected troubles, for Uranus is in retrograde this week. Ladies and gentlemen, if Uranus has given you unexpected troubles, please go and see a doctor. There is a cream for that. So look, I might not believe in horoscopes, but I do like this idea of fate. I like the idea that if we know what's coming for us, we can do something about it with science. What kind of science is that? That is the science of biology. I'm a biologist. That just simply means that I study life. More specifically, I'm a molecular biologist, which if you ask the internet means that I am really small. But actually what it means is that I'm interested in life at the smallest scale. I like really small stuff. Don't tell my partner I said that. But I really love cells, and I love the stuff that goes on inside cells. The things that govern them and kind of dictate how they interact with each other, and that's down to something called molecules. But what are molecules? Really simply put, a molecule is just two or more atoms that have been shoved together that perform a job of some kind in a chemical reaction or a process, and they pretty much govern life on Earth. That makes them, in my opinion, extremely molecule. Now, as I said, I am a PhD student and I'm a molecular biologist, so I do have a couple of favourite molecules. Number one is caffeine, very closely followed by ethanol. But in all seriousness, molecules are amazing. They're tiny, but they're mighty, and they make our world go round. And I kind of think of them as Earth's little superheroes. And there's one molecule in particular that I think is the most super of all. And that molecule is clustered regularly interspaced short palindromic repeats. Snappy, hey? Only his mum calls him by his full name. He tends to go by CRISPR. Now, when I say CRISPR, I do not mean this. I do not mean this, although I am getting hungry. What I mean is this guy here. Now, he is actually pretty big for a molecule. Science calls him a macromolecule, but I call him my hero. You can tell that he's really cool because he's got this awesome kind of glasses and hat sitch going on. But no superhero cape, because let's be realistic, he's a molecule. And look, I'm not going to insult your intelligence any further. This is clearly a cartoon, right? Allow me to show you a real-life picture of CRISPR-Cas9. There he is. Maybe this will help. Or I put his hat back on? Look, don't feel bad. He might be a macromolecule, but he's still absolutely microscopic. You have to be a fellow tiny superhero like Ant-Man to see him. So let's go back to our very helpful cartoon. You might have noticed he has another accessory. CRISPR is holding some scissors. That is because CRISPR's superpower is that he cuts DNA. Yes, you are looking at a genetic engineer, my friends. Better update the accessories. So CRISPR can cut DNA. So CRISPR can cut and change DNA. He can change the book of life because that is what DNA is. The DNA that's in all your cells contains the instructions to make you, you. It's a bit like a code or a blueprint. And it's incredibly complex, and yet somehow this code has been written with just 4 letters. Adenine, guanine, cytosine, and thymine. Or scientists like to call them A, G, C, and T, because we are very lazy. It is also why I'm saying DNA and not deoxyribonucleic acid every 3 seconds. And even though there's only 4 letters, we get these amazingly complex things like us, and that's all down to how these letters are arranged. You see, most of our DNA is arranged in blocks that's known as genes. Not that kind. That kind. So a gene is just a sequence or a string of DNA that is read by the cell, and it results in the production of a particular protein that does a specific job in a specific place. So if this, for example, we've been talking a lot about guts and colons tonight, let's say this is a gut gene. It might result in a protein for digestion. You have genes for your eyes that give you proteins that are required for vision. You have genes that are expressed in your brain that give you proteins. So there's a lot of variety on Earth in life, and it's quite amazing because we get all of this from just 4 letters, right? So how do we get all of this variety? Well, it just turns out that we have an absolute crap ton of DNA, people. In every single one of your cells, you have 6 billion letters of DNA. You know, if you took all of the DNA out of all of your cells and you laid it out in a line, it would stretch from here to the sun and back 61 times. And you'd be dead. But it is still a cool fact. So we have all of this DNA. It might be surprising to you to learn, therefore, that genetically, humans are 99.9% identical. Yes, I am very happy to tell you that you are 99.9% genetically identical to these fine specimens. Did anybody go? Oh, I did not go because I am in my poor PhD student era. You are also nearly genetically identical to these individuals. So hopefully it's obvious that actually 0.1% of the difference, it really matters, right? It's in this 0.1% of our DNA that we see differences that give us things like our various eye colours, partially dictate how tall and short we are, and can also say whether you love the taste of coriander or think it is the spawn of Satan in culinary herb form. But it's also in that 0.1% that we find mutation, all right? And I'm not talking about the cool kind of mutants. I am talking about DNA mutation that gives us disease. Diseases like cystic fibrosis or Huntington's or the most famous genetic disease of all, cancer. So for patients with these DNA mutations, a lot of the time, their fate to get disease is written into their DNA. When I told you at the beginning of this talk that I'm interested in learning how to change that fate, and I've been trying to do that with my superhero buddy, CRISPR. But how? Well, there is something I haven't told you yet about my friend here. Do you see that thing in the middle? No, ladies, he is not just happy to see you. That thing in the middle is called a guide RNA. And you can think of it as a bit like a GPS. You see, CRISPR doesn't just cut DNA randomly. He cuts it in a specific place and is guided to that place in the DNA by the guide RNA. And this is one of the bits of CRISPR that I, as a researcher, can manipulate. I can specify that guide RNA to get him to go to any place in the DNA that I want and make a change. So there are lots of diseases, genetic diseases, that we might choose to consider tackling with CRISPR-Cas9, that we should consider tackling. But I've chosen one to study that's a little bit more obscure that you might not have heard of, because I don't like research funding. No biggest warning tease for me. But I work on something called retinitis pigmentosa. Now, don't worry about engaging those brain genes. This is not a test. You do not need to remember. But this is actually the most common cause of genetic blindness. Now, it affects a tissue in the back of your eye that's called the retina. This is a little layer of tissue that is responsible for taking in light energy and telling your brain to see. So, no biggie. And the cells that are affected in this disease are called the rod cells. You have like nearly 100 million rod cells in every eye, and that's what's affected in retinitis pigmentosa. So it turns out that the rod cells really need one of those proteins I was talking about before. It's called rhodopsin. And for patients with retinitis pigmentosa, the overwhelming majority of them have a mutation in their rhodopsin gene that turns the protein toxic. Okay? So what happens is the rods make lots and lots and lots of this protein because they need it, but it's toxic and it builds up and up and up in the cell, the little shit, and it basically kills the rod cells progressively. Okay? Now, your rod cells are really important. They are responsible for your peripheral vision and for your nighttime vision. So patients start getting symptoms like tunnel vision. They also begin to experience night blindness. And eventually their retina gets so sick that they go totally blind. And right now, there is absolutely nothing we can do to slow or stop this. This is the distinctly unfunny bit of the talk, I'm afraid. And it's so unfunny that I have spent the last 4 years of my life trying to work out if I can change the fate of patients with retinitis pigmentosa with my buddy CRISPR. And I have come up with something. So, thank you. I would like to keep give me my PhD now, but unfortunately they keep asking me to write this thesis. That's a tomorrow problem. So what I've managed to do is I've managed to design a CRISPR therapeutic that can really efficiently and specifically seek out that mutant rhodopsin gene and cut it up. Like, mess it up real good, like mafia style. And what happens is the protein doesn't get made anymore, right? The gene doesn't work, so we get rid of it. So if we can then get this to work really well in an eye, then I can actually get rid of all of that mutant rhodopsin. And if that works, then we can stop the rod cells from dying and we can stop the progressive blindness and basically turn around the retinitis pigmentosa in those patients. Does that sound a little bit like science fiction to you? Because it sounds a little bit like science fiction to me, and I'm the one doing it. But actually this is beginning to happen for different diseases all around the world. The spaceship that is CRISPR has landed. Just last year, the FDA approved the first ever CRISPR therapeutic. It's for a blood disorder called sickle cell disease. It's a really painful and life-limiting disorder. And we know that this treatment works because this woman here is Victoria Gray. She's pictured with her kids. She's the first ever patient to be dosed with this in a clinical trial. She has now been out of hospital and happy and well for 4 years. So CRISPR has changed her fate. It's changed her future, and it's changed her children's futures. I would go so far as saying that CRISPR pretty much is the future of medicine. Apologies to everybody else who's also working on the future of medicine. But I don't want you leaving here thinking that it is a silver bullet, all right? CRISPR can't do everything. I am afraid that I cannot get it to turn you into this. Do not ask. You would be surprised how often I get asked about this kind of thing. I cannot make you into this. And look, it also just can't solve every problem that we have. But what I do think it means is that for patients with genetic disease, one day they're not going to be bound to the fate that's been written into their DNA. You see, I reckon we're going to be able to go them one better than reading their horoscope. I reckon one day I'm going to be able to tell them that there is a cream for that too. I have an